I'm going to tell you a little bit about the type of research that I've been doing in my laboratory the last 30 so years. So my laboratory is actually at a biomedical research institute that's a few miles from LSU, and I lead the Adipocyte Biology lab. And so I won't go too sciencey, but there's, you have like over 300 different kinds of cells in your body, and they all do different things for you. And so the kinds of cells that I've been studying for 30 years are fat cells. The fancy word for them is adipocytes. And so most people for a long time, till about 20 years ago, thought they were just kind of inert in the sense that all they did was just store energy, and they weren't really that important in any other arena. But we know now that we've known for a couple decades now that that's not true. And these cells are also highly responsive to the hormone insulin, which is really important in glucose metabolism. And adipocytes have endocrine functions, which mean they make proteins and lipids and RNA that can communicate with all the other tissues in your body. Like there's things that are made in your adipocyte that get into the bloodstream and regulate gene expression in your liver and in your muscle and in your brain and in your kidneys. So there's a lot of capabilities that you have in these cells. And when I'm asked why have I been an adipocyte biologist for 30 some odd years, my answer always comes back to this slide. And that is, if you mess up just one of those functions of an adipocyte, you make it so that it can't store lipid, or so that it doesn't respond to this one hormone insulin, or if you change one of the over 1,000 endocrine activities that it has, you just do one of those things. One of them, you have type 2 diabetes. You have systemic metabolic dysfunction. So you can change one function in your adipocyte, and it has systemic ramifications. And so that's why I think these cells are so important. And when I went to my first biochemistry meetings, when I was a graduate student, I was doing all this signaling in molecular biology, and they would say, "Oh, your data is so interesting, but why are you studying fat cells? They're inert, they don't do anything." And then we had this huge epidemic in 1996 that coincided with the obesity epidemic. So let me just take you through this slide really, really quickly. So we're starting here back in 1958, and we're just going to 2014. And so back in 1958, there were less than 1 million people in this country that had diabetes. It represented about 1% of the population. And then you can see how this continues to go up, and it really started to go up a lot in '96 as the obesity epidemic went up as well. So this only takes you to 2014, but even back in 10 years ago, there were over 22 million people in this country that had diabetes, and now it's over 30 million people in this country that have diabetes. Now, I won't dig down too deep, but in the US, in South America, and in Europe, 85% of the people that have type 2 diabetes are obese. But the genetics in Asian populations are different, so only 50% of people in Asia who are diabetic are obese. So there's a lot of lean type 2 diabetics in Asia. But in the US and in Europe and in South America, the incidence of type 2 diabetes really does correlate with obesity. And what obesity is just excess adipose tissue. So there's no doubt obesity and type 2 diabetes are epidemics, and these diseases continue to increase. And so basic research, which is what I do on adipocytes, does enhance our understanding of these diseases and can result in the development of treatments. So that's the big picture. And so, like I said, there's no doubt that, especially in the US, that obesity is a strong driver of this epidemic of type 2 diabetes. And you should care if you have type 2 diabetes. It's the leading cause of blindness in the world. It's the primary reason that people have dialysis. so nephropathies, and it's the primary cause of neuropathy. So the clinical manifestations of this disease are substantial. Another thing that people sometimes don't know is obesity puts you at risks of lots of other diseases too. Besides diabetes, of course, it can increase your risk of cardiovascular disease. Most people know that. But we've learned in the last decade or so that at least 14 kinds of cancers, the risk for those types of cancers is greatly increased with obesity as well. And the National Cancer Institute, the NCI, is going to release pretty soon that it's not smoking that's the leading cause of cancer in this country, that it's obesity. So obviously, messing with the function of fat cells has a very substantial outcome for diseases. So too much adipose tissue is bad, but you know what? Too little adipose tissue is bad as well. So if you don't have enough fat, your reproductive axis is messed up and you can't reproduce. There's different kinds of diseases where people have a mutation in some gene, or they've taken a drug that has caused this, and they have these diseases called lipoatrophy or lipodystrophy, where they lose a lot of their adipose tissue, or the fat gets stored somewhere else in the body. Like sometimes in the old days when they started using some of the HIV drugs, some of those drugs caused a humpback, which was an adipose tissue deposit. So some of those drugs can redistribute where your fat goes. So when I started my lab, I started at a very basic place studying a signaling system that is in every single cell in your body. It's called the JAK-STAT signaling system. And so there are these kinases that associate with receptors, and they phosphorylate these proteins in the cytosol that form homodimers and all these complexes, and they go to the nucleus and they regulate gene expression. And what these proteins do, they do different things. So what they do in a hepatocyte and what they do in a skin cell and what they do in a brain cell is completely different than what they do in an adipocyte. And this pathway hadn't been studied in adipocytes, so that was the first grant I got when I started my lab, is I looked at one of the members of that signaling pathway called STAT5. And we engineered some cells so that they express STAT5, and we put them into these athymic mice because then they won't immunoreject the cells. And wherever we injected those cells, we got what we call an ectopic fat pad. So we could show that this gene and this protein were really important in the development of adipose tissue. And we took a lot of other approaches to actually show that this signaling protein and transcription factor could contribute to regulating all these different properties of adipose tissue. Its ability to accumulate lipid, its ability to respond to insulin, and it even impacted the ability of the cells to secrete a wide variety of different endocrine factors. And we also, that was kind of a premeditated approach. We also take what we call shotgun approaches or exploratory approaches. And so we just isolated the STAT5, and then we figured out all the other proteins that were binding to it. And so I was telling Tri-Beta yesterday, we actually found in one of our studies that there was this protein that's normally in every cell in your body, is in the mitochondria matrix, but we found it in the adipocyte nucleus. So we're like, maybe that was just an artifact of, the way we isolated the STAT5. But of course, we used like 10 different methods, and, we showed that indeed this protein, which can generate acetyl-CoA, was in the nucleus of adipocytes. And as I was telling the students yesterday, we could never, even though we got this published in a really good journal, I could never get this project funded because people they just didn't believe it. And I've done this myself. Like, we know now, for example, there are a lot of macrophages in adipose tissue, and I saw that like in 1991 in a slide, and I'm like, yeah, that can't be right, and I ignored it. And then, 6 years later, there's like the cover of Science. I just don't ignore things. It's been the hardest thing in my career not to just say, that can't be right. Every time I do that, I try to catch myself now, because you've got to just forget all these assumptions. Your hypothesis, you've got to test it. I'm wrong all the time, but try not to just dismiss things, which I feel like I did too much of that. But anyway, we proved from microscopy to all different kinds of methods that this protein was actually in the nucleus of fat cells, both in cells, and we did it in vivo in mice as well. Could never get it funded, and now I might go back and rewrite this grant depending on what happens, but now there's been like over 10 papers in big journals to show that this is in the nucleus of cancer cells, in liver cells, and a variety of different systems. So sometimes you just have to wait for everybody to catch up, and I think now that there's a consensus out there, we might be able to get that figured out. We also made a mouse. We do a lot of genetics in the lab where we manipulate the genes of mice. And we knocked out this gene just in adipocytes, not in any of the other kinds of cells. And we predicted it would impact growth hormone signaling because that's what this protein does. And growth hormone is known to reduce fat mass. So we thought when we knocked this down in adipocytes, these mice would actually be fatter. And they were fatter. And we published this paper a few years ago. When we knock out STAT5 just in a mature fat cell, the animals have increased fat mass, but they also have improved metabolic health. So they're completely resistant to diabetes. But the kind of fat they had was subcutaneous fat. So maybe you guys have heard of this in the news. That subcutaneous fat is that fat you can pinch, the fat on your butt. But the fat that you have, that's directly, I have some funny pictures of butt fat, but I didn't show them. There's this picture of, and I've never seen the Kardashians, but there's a picture of Kim Kardashian from like 15 years ago where she just looks very pear-shaped, and you can see all her subcutaneous adipose tissue. But subcutaneous fat, you can have a lot of it, and you can still be very healthy. Whereas if you have this visceral fat that directly surrounds your organs and is under your rib cage, it's pretty bad. And this has been known a long time. So the kind of fat you have is really important. And those mice that we made that were metabolically healthier, they totally just had increased subcutaneous fat. They didn't have any increased visceral fat. And this isn't just a phenomenon from mice. The same thing is known in humans, that you can have people who weigh 600 pounds and it's all subcutaneous fat and they're metabolically healthy. They're rare, but it can happen if they don't have any increased visceral fat. So I always, tell people that, adipose tissue deposition is like real estate, just location, location, location. So where your fat is important. Some other observations we made in these mice is at the time it was thought if you had larger adipocytes, or if your adipocytes were whiter in color, which usually means they have less mitochondria, that they were always unhealthy. And this was kind of a consensus. But our paper and now a lot of other papers have shown that bigger adipocytes are not always unhealthier. So we know that is true in mice and humans. And if the tissues are a little whiter, that doesn't mean they're unhealthy either. So, bless you. So there's lots of animal models. There used to be 10 years ago, maybe there was a dozen. But now there's a few hundred animal models where the mice are have a lot more fat, and yet they're completely metabolically healthy. Okay, so there's hundreds of these animal models now. This particular animal model was made at UT Southwestern, and it's the fattest mouse that has ever been made, and you can't give this mouse diabetes. It's so metabolically healthy. They do have to go into the animal room every night, though, and make sure that they're upright, because they can't flip over, and they'll dehydrate. So, so there is you've got to think about that if you have a fat mouse. You don't want to kill it. But so here's an example, of separating out excess fat mass from metabolic health. So there's no question that most of the time, fat mass and metabolic disease go together, right? Most of the data shows that. But I showed you, just a snapshot. But if we take out STAT5 and adipocytes, our mice had more fat. but they were a lot healthier. And that mouse I just showed you, and that, like I said, there's hundreds of other examples. So I'm gonna actually skip this part of my talk. And then just tell you, we did some studies in botanical research for a little while. I had an undergrad. This was before I moved my lab to the Pennington, and she was like, hey, there's a grant you can write to study plants. And we ended up screening plant extracts from like 450 places around the globe. And we had one that had really strong anti-diabetic activity. And we gave it to mice, and it did get rid of their diabetes, but it didn't change the fat mass at all. So, and this is true for some diabetes drugs. Some diabetes drugs, they take your diabetes away, but you also lose weight, like all these drugs like Ozempic, Mounjaro, you get weight loss and you lose the type 2 diabetes. But the point I want to tell you right now is that you can improve the function of your fat cells without losing the fat mass per se. You can improve the function of those adipocytes. And we also know, and this is definitely more true in Asian populations, you can have a lot of lean individuals who are very unhealthy. And this is, typically due to problems in their liver, but it can be other tissues as well. So just because you're lean doesn't mean you're metabolically healthy. And then this study was one that really, I've exercised my whole life. I still do. And this study that came out from Harvard in 2015 really piqued my interest. So they took these mice, and if you give a mouse a wheel, it will run all night long. You don't have to force it. So this wasn't forced exercise. So they took these mice that were either sedentary or had access to a wheel for 11 days. So, and they're nocturnal, so they run at night. And then after the 11 days, they sacrificed the mice and they took out the tissue, either from the sedentary mouse that didn't have a wheel or the exercise mouse. And they transplanted that fat. I know that sounds horrible, right? Who wants more fat? But this is what they did. They transplanted the fat into diabetic animals. And the animal that got the sedentary fat, no effects. But the animal that got just the fat, the only difference is it had a fat transplant from an exercise mouse. Its diabetes went away for 3 weeks. So this shows you how powerful what's being made in that fat cell was fixing the metabolism in the whole animal. So when this came out, everyone's like, this is too good to be true, right? That exercise, we know how good it is for your mental health, for your cardiovascular health, for muscle metabolism. Is it really having very prominent effects on adipose tissue as well? Yes, it is. So this group is this was repeated by a big exercise lab at McGill University in Canada. So exercise remodels your adipose tissue. And so, sometimes people lose weight with exercise, but mostly they don't. Exercise is a great thing for weight maintenance. But that doesn't mean you should do it because it's excellent for metabolic health. People who are type 2 diabetic, even in the absence of weight loss, can reverse their type 2 diabetes if it's in the first 5 to 10 years just by exercising. And we do understand these pathways of how exercise enhances insulin sensitivity. So the last thing I'm going to tell you is what we're working on right now. We're studying this gene called CAT8, which is one of these epigenetic proteins, and it's in all cells. And we thought maybe it had an important role in adipocytes. So the problem when you study something which is like above the DNA, epigenetics, is that it has a lot of developmental effects. So all the animal mouse models we've made through the years, they're all congenital, right? So as soon as that mouse is born, that gene isn't there. But we made an adult inducible knockout. So the genetics of this, I can barely keep track of. I have to have someone else do this part for me. But we get these mice, and we can put something in their diet in an adult mouse, and we can knock out this gene just in adipocytes. And so when we knock out this CAT8 just in the adipocytes of an adult animal, you can see, and we've done it in females too, because it turns out it's important to study females as well. And they lose all their adipose tissue. You think, oh, that might be good, but they get unbelievable diabetes. They're like one of the most diabetic animal models that have been made. So you can make lean mouse models that are very unhealthy. So I just want you to have that take-home message that, you know, it's absolutely true that typically more adipose tissue is going to put you at great risk for metabolic diseases and cardiovascular diseases and different types of cancers. But you can have excess fat and improve your fat cell function, definitely by exercising, but eating well and not eating processed foods also helps quite a bit. So right now, kind of all the studies, I just told you about one project we have going on in the lab now, but all our studies right now seem to be on, you know, molecular pathways that are uncoupling fat mass and metabolic health. And here's my last slide. This is my current lab. This was at the holidays. So I have 2 research associates, Anik and Allison. Anik and Tammy, and then a senior scientist, Allison, who's been with me for 15 years, and then David is a PhD student from Ecuador, and Lindsay is a first-year PhD student from Alabama, and Courtney is from Minnesota. So I've got to work at least 4 more years so these people can finish their PhD. And yeah, that's it. Thank you very much for your time.